It may seem counterintuitive, but every day that you've barely been able to get out of bed and every time that you've struggled to keep your eyes open while cramming for a test at 1 a.m., you've actually had a lot of energy. I'm not talking about motivation or enthusiasm, I mean real physical energy. We might be used to energy as a big picture concept. Having energy allows us to move our bodies and do work. That's totally true, but cells also need energy to move their body, manufacture new proteins, and make chemical reactions happen. And that's the focus of this episode, energy at the very tiny level. Today, we're going to learn how we turn the molecules from food into usable energy. So what do we mean when we say that energy is important? Well, some of our biological processes require energy to turn reactants into products. Chemistry terms for the chemicals you start with and the chemicals you end with. Our bodies have a few ways of doing this, namely extracting energy-rich molecules from the food you eat and turning it into energy. That's where a molecule called adenosine triphosphate, or ATP, comes in. As the name implies, this molecule has 3 phosphates. But it's the bonds between them that we're more interested in. The chemical bonds that hold those phosphates together hold a lot of energy. When one of those phosphates is broken off, that ATP becomes ADP, or adenosine diphosphate, plus 1 loner phosphate. That transformation of ATP to ADP results in usable energy that our cells can use to power our biological processes. So that begs the question, where does ATP come from? Well, when an adenosine and a triphosphate fall in love, I'm kidding. Our body's main way of making ATP involves using carbohydrates, especially a simple but important molecule called glucose. Try not to think of carbohydrates as mini tortillas floating around in your cells, but as what they really are. Molecules of carbon, hydrogen, and oxygen, hence carbohydrate. The first thing we do is put that glucose molecule through the process of glycolysis. Glyco for sugar, and lysis because we're breaking it apart. That glucose molecule gets broken down into another molecule called pyruvate, plus another molecule that becomes useful a little later on. Glycolysis actually takes a little bit of ATP to happen, but it ends up netting us 2 ATP molecules, which is awesome. We will definitely use those 2 ATP. This entire reaction happens without oxygen. It's anaerobic. But our bodies can extract way more ATP if they use an additional aerobic pathway, meaning they do use oxygen. Some simple organisms, like the bacteria that causes botulism, can fuel their entire existence off of anaerobic pathways like glycolysis. But in our bodies, glycolysis is just the first step to cranking out a lot of ATP. By the end of glycolysis, we have 2 pyruvates, 2 ATP molecules, and 2 molecules of NADH, a molecule that we can't extract energy from, but we can repurpose as ingredients in a different reaction. Now, glycolysis happens in the cytosol, the liquid within your cells. But like we learned in the last episode, we've got a secret weapon, a behemoth of energy production. We have mitochondria. I know, we spent so much of the last episode talking about mitochondrial DNA and endosymbiosis, and today we finally get to talk about its powerhouseness. I'm excited too. Okay, let's go. So after glycolysis, we shuttle that pyruvate and those NADH towards the mitochondria, where we're gonna make even more ATP. After all is said and done, we're gonna end up with more than 30 ATP. Our cells like to work smarter, not harder. So they use molecules called enzymes. Enzymes are chemicals that lower the amount of energy required for that reaction to happen. I like to think of enzymes as coupons for chemistry. You get the same product in the end, but instead of spending a lot of energy, you apply an enzyme and you get the same product for a lot less energy. And this next process, the Krebs cycle, has multiple enzymes helping it along. Wait, don't click away yet. Look, I get it. I've had to memorize this thing 3 separate times throughout my schooling because I kept on forgetting it. That's the real Krebs cycle. You learn the Krebs cycle, then you forget the Krebs cycle, so you learn the Krebs cycle, and then you're spiraling forever in the nerdiest episode of Black Mirror ever written. But once you see the big picture of this cycle, you'll come to appreciate it as I have. Remember, the purpose of all this is to make ATP. And we can make a lot of ATP if we can use oxygen. But we need to prep our materials in such a way that lets us use oxygen. Pyruvate itself has 3 carbon atoms. Along comes a chemical that bumps one of them off, turning it into a molecule with 2 carbon atoms. This new product is called acetyl-CoA, and it's a big deal in the Krebs cycle. Next, we add a 4-carbon molecule to acetyl-CoA to make a molecule with 6 carbons called citrate. The Krebs cycle is also called the citric acid cycle because of this molecule. So by this point, we have a modest 4 ATP, 2 from glycolysis and 2 more from the Krebs cycle. But the more interesting products are the 10 molecules of NADH and the newly created 2 molecules of FADH2. These things are really going to pay off in the next step. Manipulating these leftover ingredients will get us a lot of ATP. It's a process called oxidative phosphorylation. Again, big science words, but it means exactly what it says. We're going to shuffle around some phosphates and use oxygen. In order to contain and process that energy, the NADH and FADH2 molecules transfer their electrons along a series of steps called the electron transport chain. They present their electrons to the mitochondria's inner membrane, where electron transporters move them towards the inside of the mitochondria. This process releases some energy, which sets up a smooth gradient of hydrogen ions across the mitochondrial membrane. This gradient can be used to power ATP synthase, an enzyme that helps put a phosphate on ADP, turning it into ATP. After you tally everything up, you realize how much energy you generated. You gained 2 ATP directly from glycolysis and 2 more from the Krebs cycle. Each of those 10 NADH molecules can get us up to 3 ATP, and each of those 2 FADH2 can yield 2 ATP. That's a total of 38 molecules of ATP for every molecule of glucose under prime condition. You also ended up with water and carbon dioxide as byproducts. But how cool is that? We went from 2 ATP per molecule of glucose to 38 by adding oxygen and a few enzymes. Now, we can extract ATP from a few other molecules. Glucose itself is a simple carbohydrate, but we can also utilize more complex carbohydrates by breaking them into simpler versions. Then they go through a similar process to before: glycolysis, Krebs cycle, oxidative phosphorylation. Of course, it would be great if we just always had a batch of glucose on hand to use whenever we needed it. Like, we always have some dissolved glucose in our blood that our cells can use, but it's constantly increasing or decreasing depending on things like food, exercise, time of day, or whether or not you have diabetes. And low blood glucose can get dangerous, especially if your brain doesn't have enough glucose, since that's the only fuel source it can use. Unless you're starving. As a workaround, our bodies convert spare glucose into an easily usable storage form of glucose called glycogen. This starchy substance is kept mostly in the liver and skeletal muscle and can be tapped whenever your body needs some quick energy. Your liver can sense when overall blood glucose is low and chip off some glycogen to get used as fuel. And your muscles can use glycogen if they need energy during exercise. Now, stored fat is a more energy-dense molecule than glucose. However, fat is more than just energy storage. It's a whole organ unto itself. But for now, we'll focus on how we extract so much ATP from it. When our bodies want to use some of that stored fat, it first needs to break it into fatty acids and glycerol. By the end of its processing, we end up with some familiar ingredients: acetyl-CoA, NADH, and FADH2. Then that acetyl-CoA goes into the Krebs cycle, just like if it came from a glucose molecule. However, what makes using a molecule of fat different than a molecule of glucose is the total ATP payoff. For a typical molecule of fat with 16 carbons, we'll end up with 21 molecules of ATP from NADH, 14 from FADH2, and 96 from acetyl-CoA for a whopping total of 131 molecules of ATP. Although, caveat here, this isn't a concrete number since fat comes in multiple carbon combinations. Either way, it's a lot of ATP. Now, molecules of fat are bigger than molecules of glucose, but a gram of fat still nets you 2.5 times more ATP than a gram of glucose. That's what makes it such a great way of storing energy. But that's only one of the amazing functions of our fat. In the next episode, we're going to talk about fat as an organ and learn about all the other things it does aside from storing energy. Thanks for watching this episode of Seeker Human. I'm Patrick Kelly.